Hello friends, welcome back to my YouTube channel Nesting Padasale. So we are continuing the system physiology animal notes. So this is the part 4th video. So today we are going to study the important topic coming under digestive system and excretory system. So first topic is absorption in small intestine. So how different component of the nutrient are being absorbed by the small intestine. So vitamins and dietary lipids will be absorbed by the process of simple diffusion, whereas your protein and vitamin B12 will be absorbed by active transport. Fructose will be transported by facilitated diffusion process. Next, your monosaccharide are absorbed by facilitated diffusion and active transport. Next, your glucose and galactose are absorbed by secondary active transport. So next we will be studying about Na plus glucose symporter. So the transport of glucose and galactose depends on the presence of sodium ion in the intestinal lumen. Okay, so if the intracellular mean within the cell. Okay, so if the intracellular concentration of the sodium ion is very low in the intestinal cells, means Na plus ion will move into the cell. So here you can able to see a detailed mechanism of all the process, friends. So okay, so glucose, when Na plus move into the cell, this glucose will also move into the cell along with the Na. So here you can able to see, so this is a intracellular space and this is a intestinal lumen and this is a enterocytes, which are digestive system cells. Okay, so here you can able to see these are the tight junction. The main mechanism of tight junction is to prevent the backflow of the glucose. Okay, so here this particular thing is called as GLUT, that is glucose transporting protein. Okay, next this protein is called as, this is actually Na plus glucose symporter protein, friends. So please make a note, it is a symport mechanism, not an antiport mechanism. Okay, next this is a sodium potassium pump which will involve in transporting sodium ion and potassium ion. Okay, so in the intestinal lumen there will be low glucose and high volume of Na plus, whereas in enterocytes there will be high glucose concentration and low Na plus concentration. Okay, so glucose transport is absolutely an example of secondary active transport mechanism because the energy of uphill transport of glucose. So here in the enterocyte the concentration of Na plus is very low, whereas the concentration of glucose is very high. Okay, so the energy for uphill transport of glucose comes from the downhill transport of sodium ion. Next, the glucose, that is glucose diffuse out of the cell by a facilitated diffusion mechanism by a transporter protein called GLUT transporter protein. Next, these tight junction will block the backflow of the glucose. Okay, so this is an important concept that you should know. This is a mechanism of how your sodium glucose symporter is being working. So glucose will not able to transport outside of the intracellular space, friends, by their own. They need a help of the Na plus ion. So that's how they are using this sodium glucose symporter. Okay, so after the mechanism, then glucose will be transported by a mechanism called facilitated diffusion. So next, mechanism of glucose uptake by the cell in the presence of insulin. So insulin lowers the blood glucose level by accelerating the transport of glucose into the cell. So glucose transport between the cell and the blood is done by a plasma bound carrier protein called as glucose transporter, that is GLUT. Okay, next thing, 14 isoform of this GLUT is present, but only GLUT4 is the only transporter that will respond to the insulin. So this GLUT4 is not present in the plasma membrane in the absence of insulin. It is present in the plasma membrane only in the presence of insulin. Next, this GLUT will be stored as an intracellular vesicle. So upon insulin stimulation, it will fuse with the plasma membrane, thereby increasing the sugar uptake several fold. So let me explain you. So this is a condition of absence of glucose and this is a condition of presence of glucose. In the absence of glucose, the insulin receptor is being empty, there is no glucose and glucose ions are surrounding outside. Okay, so in the cytosol, the GLUT4, which is this particular thing is called as GLUT4. GLUT4 will be bounded to a secretory vesicle. Okay, so once the insulin is being present, then insulin will bind to your insulin receptor, then the secretory vesicle will start to bound to the plasma membrane and it will form the receptor over here. So now the glucose will bind and it will be involved in sugar uptake. Next, the regulation of glucagon and insulin secretion. So the principal mechanism of glucagon is to increase the blood sugar level when the blood sugar level falls below normal. Next, insulin helps to lower the blood glucose level when the blood glucose level is high. Okay, and the level of both the glucose as well as, that is glucagon as well as insulin, is made by negative feedback mechanism. That is, first I'll tell you glucagon, then we'll study insulin. First, low blood glucose level is now called as hypoglycemia. In that particular condition, the alpha cells will be stimulated to secrete glucagon. So glucagon will be acting on hepatocyte to induce a process of glycogenolysis and gluconeogenesis. So let me explain you. Glycogenolysis means glycogen will be break down into glucose 1 phosphate and glucose. Next, this glycogenolysis occurs in hepatocyte and myocytes. Next, the key enzymes which is involved in the process of glycogenolysis is phosphorylase kinase and glycogen phosphorylase. Next, gluconeogenesis. So it transforms a non-hybrid carbohydrate substances like your lactate, amino acid, glycerol into glucose, and this particular mechanism occurs in kidney and liver. Next, the regulatory enzymes involved in this process is pyruvate carboxylase and phosphoenol pyruvate carbokinase and malate dehydrogenase. The next process is the glycogenesis. Okay, so it is a process of glycogen synthesis in which the glucose molecules will be added to chains of glycogen for the storage. So it is a storage process and it is involved in process of glycogen. So both the glycogenesis and glycogenolysis is an opposite and antagonistic effect. So glycogenesis involved in synthesis of glucose, glycogenolysis involved in breaking of glycogen into glucose. Okay. Next, this particular process will be taking place when the blood glucose level are sufficiently high, okay, to allow the excess glucose to be stored in liver and muscle. Okay, the key enzymes are glycogen synthase, which is also called as UDP, glucose glycogen glucosyl-transferase. Okay, this is the key enzymes involved in this process. Next is lipogenesis. Lipogenesis is a process of converting your fatty acid and glycerol into fat. Okay, so in a more technical way, it is a process of in which acetyl-CoA is converted into triglyceride for the storage in fat. Then this process occurs in liver and adipose tissues, and enzymes involved in lipogenesis are acetyl coenzyme A carboxylase, that is ACC, fatty acid synthase and ATP citrate. So coming back to the point, so this insulin acts on various body cells in order to accelerate the diffusion of glucose into cell. It also increases the glycogenesis and lipogenesis. So here glucagon induces the glycogenolysis and glucogenesis, which these two processes liberate glucose. So that's how the blood glucose will be increasing. Whereas here this particular insulin will increase, that is induce a process of glycogenesis and lipogenesis. Both are involved in converting the excess glucose into fat. Next, this insulin also slows down the process of glycogenolysis and glycogenesis. Okay, so then the blood glucose level will fall. So here glucagon, after the inducing glucose released by hepatocyte, raises the blood glucose level to normal. Next thing, this is a negative feedback mechanism, friends. So if the blood glucose continue to rise, means then hyperglycemia condition will be result, which will ultimately inhibit the release of glucagon. Whereas here, ultimately after the blood glucose level falls, so if the blood glucose continues to fall, then the hyperglycemia condition will be created, which will be inhibiting the release of insulin. So next thing is the glucagon and insulin. So what I explained you there, I just given as a tabulated column. So this glucagon stimulate the glycogenolysis and glucogenesis, whereas insulin inhibit both the process. Next, this glucagon inhibit the glycogenesis and glycolysis, whereas insulin will stimulate both the process. Next, this glucagon will increase the glucose production from liver, as well as it increase the blood glucose level, whereas this insulin inhibit both the process. Okay. So next, with respect to triacyl glycerol, hydrolysis of this triacyl glycerol, glucagon will increase the triacyl glycerol synthesis, that is hydrolysis, whereas this insulin will decrease the triacyl glycerol hydrolysis. This is a hydrolysis process, not a synthesis process. Next is your lipogenesis. So glucagon will decrease the lipogenesis process, whereas your insulin will increase the lipogenesis process. So next is exocrine cells. So exocrine glands secrete a substance into a ductal system to epithelial surface, whereas your endocrine glands will secrete the products or hormone directly into bloodstream. Next is endocrine gland comprises of two important divisions. One is acnes and duct with a different cell type. So there are four important cell types. First one is serous cell, which will secrete a protein of an enzyme. So example include a gastric chief cells and tiny cell. Next is mucous cell, which will secrete a mucus. Example include Brunner's gland, esophageal gland and pyrolic gland. Next is seromucous gland, which will secrete both the protein and mucus. Example includes a salivary gland. Next is sebaceous gland, which will secrete a sebum, which is actually a lipid product. So these glands are also called as oil gland. Example include Fordyce spots and Meibomian gland. So the next two important process exocrine glands is exocrine cells include a chief cells which is also called zymosin or peptic cell, which secrete a process compound called pepsinogen and gastric lipase, whereas this parietal cell which is also called as oxyntic cells. So one time CSIR has asked the question with respect to this type of cells. Okay, so it secretes a HCl which is a gastric acid, as well as it will secrete the intrinsic factor. So first HCl, it will mechanism or function of HCl is to activate the pepsinogen to its active form called pepsin by cleaving of the small fragment. Okay, this HCl also denature the protein and kills off most of the microorganism that has been ingested along with the food. Next is the intrinsic factor, which is important for the absorption of the vitamin B12. Okay, so if the intrinsic factor is not present, like if the parietal cells doesn't synthesize this intrinsic factor, means then vitamin B12 will not be absorbed. So ultimately the RBC production will be decreased, and this is how the perinicious anemia result. Next is your enteroendocrine cell. Though there are different types of enteroendocrine cell. These are nothing but these are the cell which are present in the gut, digestive system and pancreas. First is K cell, which will be secreting a gastric inhibitory peptide called as incretin, and the function of this incretin is triglyceride storage, and it will stimulate the insulin secretion, and this incretin will inhibit the gastric acid secretion and motility. Next, L cell will secrete different compound. First one is that it will be secreting glucagon like peptide and incretin compound and peptide YY3 to 36 peptides. Okay, next oxyntomodulin, next glucagon like peptide 2. Next, these L cell are primarily it will be found in the ileum and large intestine, but some are also found in duodenum and jejunum. Next is I cell, which will be secreting the CCK, which is present in the duodenum and jejunum, and the function of the CCK secreted by I cell include they modulate the bile secretion and exocrine pancreas secretion and satiety. Next thing is that you stimulate the pancreatic enzyme secretion. Next you stimulate. Next it will lead to a contraction of gallbladder. So once the gallbladder contracted, then the bile juice and gallbladder will start secreting lots of juices that helpful for the digestion. Next it will also lead to relaxation of sphincter of ODT, which is nothing but a muscle which is involved in digestion and absorption of all nutrients. Next G cell will be secreting a gastrin. This gastrin will be stimulating the gastric acid secretion, that like your HCl, that is hydrochloric acid and pepsinogen will be secreting, and it will be increasing the movement of stomach, whereas this the G cell will increase the movement of stomach, that is motility, whereas K cell is inhibiting the motility. Next is enterochromaffin cell, which will secrete the arterial natriuretic peptide and serotonin. Next, enterochromaffin like cells, which is a neuroendocrine cell, which will secrete a histamine neurotransmitter. Next, your N cell, which is located in jejunum. So N cell is releasing a neuropeptide called neurotensin, and the function of N cell is that it will control the smooth muscle contraction. Next, the S cell, which is secreting secretin, and it generally secretes the secretin from duodenum and jejunum. Next, the function of S cell include it will stimulate the exocrine pancreas secretion, and it will increase the secretion of bicarbonate by the ductal cells of pancreases and biliary duct. Next, this S cell will decrease the gastritic secretion. So the G cell, it will increase the gastric acid secretion, whereas S cell, it will decrease the gastric acid secretion. Next is your D cell, which is also called as delta cell. So D cell will secrete a paracrine signaling growth inhibiting molecule called somatostatin. Okay, so the D cell again, as similar to S cell, it will inhibit the gastric acid secretion and your pancreatic exocrine secretion, and as well as the motility of stomach. So this is how this is the important topic coming under digestive system. Next, coming to excretor system. First, we'll study the hormone or autocoids that influence the glomerular filtration rate, that is GFR. Okay, so if you take norepinephrine, epinephrine, endothelin, angiotensin, means these hormone will actually decrease the glomerular filtration rate. If you take the endothelin, epinephrine and all, they are released from the adrenal medulla, and they are generally involved in constructing the afferent and efferent arteriole of glomerulus. Okay, next, your endothelial derived nitric acid and your prostaglandins will increase the glomerular filtration rate. Next, the hormone. Next, we'll study the hormone that regulate the tubular reabsorption. So these are the list of hormone, friends. So first is aldosterone. The site of action is collecting tubule and duct. Angiotensin 2, which the site of action of angiotensin 2 is proximal tubule. Thick ascending loop of Henle, ascending loop of Henle, distal tubule and collecting tubule. Whereas antidiuretic hormone and arterial natriuretic peptide, both at the site of action is same. That is distal tubule and collecting duct. Next is a parathyroid hormone, which generally act on proximal tubule, thick ascending loop of Henle and distal tubule. Whereas the effect include aldosterone will increase the sodium reabsorption, water reabsorption as well as potassium secretion. Whereas this angiotensin same, it will increase the sodium absorption, reabsorption and water reabsorption as well as it will increase the H plus ion secretion. Next is antidiuretic hormone, it will increase the water reabsorption, whereas this arterial-natriuretic peptide which will decrease the sodium reabsorption. Next your parathyroid hormone which will increases the calcium reabsorption in kidney. If you take this arterial-natriuretic peptide and aldosterone and angiotensin, both are having opposite effect. So aldosterone and angiotensin are increasing the sodium reabsorption, whereas your ANP is decreasing the sodium reabsorption. Next we'll study in detail. First one is angiotensin 2. So this angiotensin 2 affect the renal physiology in three main ways. First, it decreases the glomerular filtration rate by causing the vasoconstriction of the afferent arteriole of glomerulus. So you know glomerulus is a part of the nephron, and nephron is a basic structural and functional unit of kidney. Next is angiotensin 2 will enhance the reabsorption of sodium chloride water in a proximal convoluted tube. Next, it will also stimulate the adrenal cortex which is present on the top of kidney to secrete aldosterone hormone, which will in turn stimulate the principal cells in the collecting duct in order to reabsorb more sodium and secrete more potassium. Next is there vasopressin, which is also called as antidiuretic hormone. This is the hormone which is released by the posterior lobe of pituitary, and the water reabsorption it is responsible for water reabsorption in distal convoluted tubule and collecting duct of nephron. Say the person didn't drink enough. If a person didn't drink enough water means the posterior lobe will releases the antidiuretic hormone which will cause more water to get reabsorbed. As a result, the less urine will be formed. So this is a process of antidiuresis. Next, if the person drink more water and does not urinate, that is if the person drink more water and does not perspire means then antidiuretic hormone will not be released. Okay, as a result, the water reabsorption will not be occur. As a result, what will happen? More water will be excreted. As a person, the person will form more urine. This is a process of diuresis. Okay, so this vasopressin generally increases the amount of solute free water reabsorption back into blood circulation from the filtrating kidney tubule of nephron. So let me explain you. So this is how the mechanism of this vasopressin or ADH. So this is a tubular remain, this is a perivascular cell lining of late distal tubular collecting duct, this is intestinal fluid, and this is a peritubular capillary. Peritubular capillary means these are the tiny blood vessels present in your kidney. So in the kidney, ADH is present in the water present in more quantity, friends. So this is what when the ADH is present in blood means it will bind to the receptor of late distal tubular collecting duct. So the ADH is present means this ADH receptor will be present in the principal cells of the distal tubular collecting duct. So okay, so after binding to the ADH receptor, this binding will activate a cyclic AMP which is a secondary messenger which will ultimately increases the secretion, insertion of water channel into the membrane. So this aquaporin are nothing but they are the water channel protein, friends. So once the ADH receptor has been connected to the ADH means that is once the vasopressin hormone has been fixed to its receptor, it will activate the water channel to get merge or to get connected with the plasma membrane by the process of exocytosis. So the next thing is that in the membrane, this membrane will be impermeable to water. So only in the presence of ADH, this membrane will be permeable and allowing the entry of water from the kidney to the blood. If this particular ADH is present, then it is impermeable to water. So once after the insertion of AQP, that is aquaporin 2, it will be permeable to water. Then what? The water from the kidney will be transferred into the bloodstream by a protein called aquaporin 3. So you should know the function of aquaporin 2 and aquaporin 3. So aquaporin is nothing but these are the water channel or channel protein that are made up of a large family of intrinsic protein. Okay, so the main mechanism is the facilitating the transport of water between the cells. And with regard to structure, they have a 6 membrane spanning alpha helical domain. So with both carboxylic and amino terminal end. Okay, in special there is a 2 hydrophobic loop that contains a conserved asparagine proline alanine, that is NPA motif is present in aquaporin. Next, these are the 4 type of aquaporin. All are present in kidney only. 1 and 2 protein is present apical side, whereas 3 and 4 is present in basal side. Next, aquaporin all are responsible for reabsorption, whereas this aquaporin 2 is responsible for water reabsorption in response to ADH, whereas aquaporin 3 is responsible for water reabsorption. In addition to this, the aquaporin 3 is also responsible for your glycerol permeability into cell. And here they had given the location of aquaporin 1. Aquaporin 1 is present in the proximal convoluted tubule and the thin descending loop of Henle, whereas aquaporin 2 is present in connecting tubule, cortical collecting duct, outer medullary collecting duct, inner medullary collecting duct, whereas aquaporin 3 is present in collecting tubule, cortical duct, and it is only present in outer medullary collecting duct, not in inner medullary connecting duct. Next is aquaporin 4 is present in inner medullary connecting duct. So next, the hormones that is we are continuing the hormones that is involved in regulating the tubular reabsorption. First is arterial neuritic peptide. So an increase in blood flow to arteria, that is arteria which is present in heart, can able to releases this particular hormone, and this particular peptide will cause a vasodilation, which is ultimately decreasing the blood pressure. Okay, so vasodilation occurs means the decrease in blood pressure occur. If vasoconstriction occur means the increase of your BP will occur. This ANP also inhibit the reabsorption of sodium as well as water, which will increase the urinary excretion, which helps to return the blood volume back to normal. So the next thing is the regulation of this ADH secretion or antidiuretic hormone secretion. So the agent or the factor which will increases the ADH include, if the blood plasma osmolality is in increased condition means, then it will secrete this ADH, which will lead to water reabsorption, and it will decrease the osmolysis of the plasma. Next, decrease in blood volume and blood pressure will increase the release of ADH, which will do a vasoconstriction to increase the blood pressure. Next is decrease the compound which will decrease the ADH include decrease in plasma osmolality and increase in blood volume and blood pressure. In addition to that, nausea and hypoxia will increase the ADH secretion. With respect to drugs, your morphine, nicotine, cyclophosphamide will increase the ADH, whereas drugs like alcohol, cholinidil, haloperidol, which is a dopamine blocker, and this particular thing is an antihypersensitive drug, will actually decreases the secretion of ADH. Next, the control of thirst. So the component which will increase the thirst so that you can drink more water include increase in plasma osmolality, increase in angiotensin II hormone, and decrease in blood volume and blood pressure. Next, the component or factor which will decrease the thirst include decrease in plasma osmolality and increase in blood volume, increase in blood pressure, and decrease in angiotensin II. Next, we'll study what are the factors that can alter the potassium distribution between the intracellular fluid and extracellular fluid. First, we'll see the factor that shift the potassium ion into the cell. That is when this are the factor which will be shifting the potassium ion, that is decrease extracellular potassium concentration. This include insulin, aldosterone, beta adrenergic stimulation, and alkalosis. Next, the factor that shift the K plus out of the cell include your insulin deficiency, that is in the condition where there is an increased extracellular potassium concentration, friends. So insulin deficiency, that is in the cases of diabetes mellitus. Next, aldosterone deficiency, that is in the case of Addison disease. Then beta adrenergic blockade, acidosis, cell lysis, strenuous exercise, increase in extracellular fluid osmolality, all will shift the K plus ion out of the cell. Next, we'll study about the diuresis, the mechanism of action, and tubular site of action. So diuretic is nothing but these are the water pill, just like your tablet or medication, that will be designed to increase the amount of water and salt expelled out of the body as urine. So there are different types of diuretics present, friends. So osmotic diuretic, example is mannitol. The mechanism is that it will inhibit the water reabsorption, solute reabsorption, generally by increasing the osmolality of the tubular fluid. And the tubular site of action include mainly these osmotic diuretic like mannitol will be acting on the proximal tubule. Next is loop diuretics, and the example include furosemide and bumetanide. Okay, so they will inhibit the sodium potassium chloride co-transport in the luminal membrane, and the site of action is the thick ascending loop of Henle. So you should know whether the mechanism is occurring in thick ascending loop or thin ascending loop. Next is the thiazide diuretic, which include hydrochlorothiazide and chlorothalidone. The mechanism is that it will also have same inhibiting, only just here the loop diuretic will inhibit the sodium potassium chloride, whereas this thiazide diuretic will inhibit the sodium chloride co-transport in the luminal membrane, and they generally act on distal tubule, whereas this aldosterone antagonistic and sodium channel blocker will be acting on collecting duct, whereas this carbonic anhydrase inhibitor will be occurring on proximal tubule. Your osmotic diuretic and carbonic anhydrase inhibitor will be acting on proximal tubule. Okay, so if you see the mechanism of this carbonic anhydrous, they will inhibit the H plus secretion as well as bicarbonate reabsorption, which will lead to a reducing, which will ultimately reduces the potassium reabsorption, not the potassium absorption. They will reduce the potassium reabsorption from the kidney. Next, the aldosterone antagonistic, they will inhibit the action of aldosterone tubular. So previously we dealt with the what are the mechanism of aldosterone. So ultimately it will decreases the sodium reabsorption as well as potassium secretion will be decreased. Next is mechanism of sodium channel blocker include, it will block the entry of sodium into sodium channel, which will be present in the luminal side of kidney, as well as same mechanism. Just like your aldosterone antagonistic, the sodium channel blocker will also decrease the release of, that is a reabsorption of sodium, and also decreases the potassium secretion. So with this we are completing the excretory system. Till only one section is available, endocrinology and reproduction. Definitely I share it with the next video, that is part 5. So with the part 5 we will be completing the system physiology animal. Next will be studying evolution. So please subscribe to my YouTube channel, only then it will be very much helpful. Thank you friends.